Well, thanks very much, and it's really a great pleasure to be giving this seminar today. I will start. So, yes, thanks everyone for coming. And as Yelena said, I'm a postdoc in Gary Lewin's lab at MDC Berlin, and today I'll be talking about Elkin-1, which we published recently. And if you want to look up the paper, you can scan the QR code here. And after that, I'll be talking a little bit about a new direction that I'm working on neuroproteomics to identify new proteins and ion channels. And I got a small grant recently on this from the European Pain Federation and the Grunenthal Bio company, basically. Okay, so let's start with Elkin-1, which we found had a profound role in touch sensation. And so we perceive our surroundings by the 5 canonical senses. Amongst them is touch. And as we all know, touch is sort of less studied, and that's perhaps because it's slightly more complex. It starts at our largest organ called the skin, which is richly innervated by a lot of different nerve fibers. And these fibers basically transduce physical forces to electrical signals. So the physical forces range from like a massive variety of physical forces that we can sense on our skin, from like a light cotton swab-like touch to heavy pinprick. And this great diversity is transduced by different nerve fibers that innervate the skin. So for example, the light end of things are transduced by rapidly adapting or slowly adapting fast conducting nerve fibers, which are highlighted here in green and blue. And the heavy pinprick-like mechanical pain is transduced by slow conducting nerve fibers called nociceptors. These nerve fibers have their cell bodies in a structure called the dorsal root ganglion, and I've highlighted it here because that will be the star of the show today. So how do we differentiate between the fast conducting and slow conducting nerves at a DRG level? And so very simply speaking, the large DRG neurons are the like the fast conducting nerve cell bodies, so they conduct light touch, whereas the small and medium DRG neurons are for the nociceptors, so the heavy pinprick or mechanical pain. After that, the information travels to the spinal cord and then to the brain. So what's happening here is we are converting physical forces into electrical stimuli. So how does it happen, right? So what are the proteins then that can do it? And this has been a question for a very long time in the field, until in 2010, the lab of Artem Pataputian characterized the piezo proteins or the piezo ion channels that can convert physical forces into electrical signals. Later on in 2014, they also showed that piezo2 is one of the transducers of mechanical forces for touch sensation in mice. So they found that this is the major ion channel in the somatosensory system. So Gary's lab collaborated in these papers where they showed that with skin nerve recording, they found that there was a profound loss of fast conducting nerve fibers mechanosensitivity. So you can see here the A beta, so the fast conducting nerves which transduces light touch, lose about 50% of their mechanosensitivity in this model where piezo2 was knocked down from the peripheral nervous system. And then in another model also, they found that when piezo2 was knocked out from the peripheral nervous system, about 40 to 50% of the fast conducting A beta fibers lose their mechanosensitivity. So then, of course, the question is, what are the other proteins that are involved in touch sensation? Because we still have quite a lot of nerve fibers that do transduce mechanical forces. So at this time, Gary's lab was working on another protein called Elkin-1, which they found, like Kate, another postdoc in the lab, found that it was involved in transducing mechanical forces in a melanoma cell line. This is sort of the time when I joined the lab. And so I sort of replicated some of the experiments from this paper. So Elkin-1 preferentially evokes mechanically active currents when there is substrate deflection. Additionally, I also transfected piezo-1 knockout cells, so hex cells. So these are the cells that do not produce indentation-induced currents normally. So as you can see in the untransfected cells, there were no currents with poking. But when I transfected these cells with Elkin-1 construct and performed poking with patch clamps, these are the poking stimuli, I could see currents. So these were not super robust currents. As you can see, not all the transfected neurons had current, but the ones that did was quite large in amplitude. So this shows that Elkin-1 has indentation-gated currents, mechanically active. And then I also did cell-attached patch clamp on these cells, transfected either mock construct or Elkin-1 construct. And during this cell-attached configuration, I gave like slight positive forces to positive pressure to these cells. And these recordings were conducted at plus 20 millivolts. So for the electrophysiologists amongst the audience, this might be a very high voltage, but this was because in another study, it was shown that Elkin-1 has a high open probability in very positive potentials. And I will talk about it very briefly at the very end. Okay, so in this configuration, upon positive pressure application, I saw that in the Elkin-1 transfected cells, the rate of events were increased when a pressure was applied. This also says that Elkin-1 can sense mechanical forces such as mild pressure. So then I started looking at the major question then, is Elkin-1 involved in somatosensory touch? And for that, the star of the show, as I said, is the DRG neurons. So I looked at single cell transcriptomics datasets that were already there to see where Elkin-1 is in the DRG neurons, because DRG neurons have a lot of different subsets, and I found that Elkin-1 was expressed in all the different subsets of DRG neurons from these datasets. This I then validated using RNA-scope and antibody staining. So what I saw here is that although Elkin-1 is largely expressed, there are about 50% of the neurons that has a very high expression of Elkin-1. All right, so then we generated Elkin-1 knockout mouse line, and I validated this mouse line again using RNA-scope and antibody staining to show that yes, indeed, Elkin-1 was knocked out from this mouse line. And I also did a variety of stainings for other DRG neuronal markers to show that deletion of Elkin-1 do not disrupt the molecular architecture, basically. Okay, so let me actually try to switch off this. All right, great. So now you can see the title a bit better, hopefully. So deletion of Elkin-1 does not disrupt molecular architecture of DRG neurons. So what then is it doing in So what is it doing in mice, right? So to look at that, I conducted a series of behavioral assays. So I wanted to see upon application of physical stimuli, how the mice behave. And these are evoked mechanical stimuli. The first thing was the cotton swab. This is basically a Q-tip that you use to sort of clean your ears. And then what I did is pulled it out to 3 times its original volume and then swept it across the feet of mice. So what happens in a control or a wild type mouse is that as soon as you sweep it across its paws, it withdraws its paws. So that's what we consider a response. But then when I did the same thing with Elkin-1 knockout mice, what I saw is 50% of the times these mice do not withdraw their paw. So these very light forces are unable to evoke the same response in Elkin-1 knockout mice when compared to control. Then I did another assay called the von Frey filaments assay, which are measured force filaments, which when applied to the paw of mice, the mice withdraws its paw. And here what you see is in the Elkin-1 knockout animals, more force, like more heavier von Frey filaments are needed for the mice to withdraw its paws. And this I also did it with a series of different von Frey force filaments to show that in all of these force bins, basically Elkin-1 mice has a deficit in withdrawal. So taken together, this suggests that Elkin-1 mice are less sensitive to physical forces, and I validated that using cotton swab and von Frey filaments. So what then is the neuronal basis of it? That was the next question. And for that, Gary devised this protocol called the electrical search protocol. So what happens here is you take the skin off of mice. So the paw, you take the skin with the nerves intact. Then you record the nerves with an electrode. At the same time, you find the receptive field of these identified nerve. And in this receptive field, you sort of give a physical stimuli, like a poking stimuli. If the nerve responds, then it means that the nerve is mechanically active. If it does not respond, the nerve is mechanically inactive. So using this protocol, Gary showed that about 30% of the A beta fibers, so just as a reminder, these are the fast conducting fibers that detect light touch. He found that about 30% of these fibers were mechanically inactive in the Elkin-1 knockout animals. So then what's going on then in these nerves that are mechanosensitive? So to look into that and characterize this further, another postdoc in the lab, Mohammed, utilized this skin nerve setup, and he showed that even the nerves that are mechanically active are actually very deficit in transducing mechanical forces. So this is an example of a slowly adapting mechanoreceptor. So what happens in a wild type scenario is that if you give indentation stimuli, so a poking stimuli, the wild type slowly adapting nerve fiber keeps firing throughout the entire stimuli, right? But what we see in the Elkin-1 knockout nerve is that it starts firing as usual, but then it basically stops. So there is a very strong adaptation perhaps, or these fibers are inherently deficit in transducing mechanical forces. So this is happening at the skin level. So I then wanted to know if this is also the case at the neuronal level, so at the DRG level, where then there are not other skin cells around. So to do that, I recorded from the large diameter DRG neurons, because these are the cell bodies of the fast conducting nerves which detect light touch. And I recorded from these neurons using patch clamp electrophysiology with poking, so indentation. So what happens is I'm recording from this here, and I'm poking the cell surface simultaneously. And if I do that, I see 3 distinct type of biophysical properties of the currents. So the first is rapidly adapting current, where the current shuts off very quickly. Then there is a intermediately adapting currents, and then there are currents which takes more than 50 milliseconds to shut off, and these are the slowly adapting currents. So just simply speaking, there is an accepted idea in the field that the rapidly adapting currents are possibly more important for light touch sensation. Okay, so using this protocol, what I found was that the Elkin-1 knockout, in the Elkin-1 knockout animals, about 40% of the large diameter sensory neurons did not respond to mechanical indentation. And the ones that were mechanically active, a large number of these neurons did not have rapidly adapting currents. So this sort of goes very well with the two other sets of data that I showed you, that the mechanoreceptors at the skin level have less mechanosensitivity, and at a behavioral level, the Elkin-1 mice do not sense physical forces that adeptly. All right, so and this data I also validated using another assay called the pillar assay, which we developed in the lab, in which you sort of plate the neurons on top of these elastomeric pillars, and then you move the pillars from underneath. So that shows that also with substrate deflection, the Elkin-1 neurons do not sense mechanical forces that much. All right, so one can then argue that, okay, this is a global knockout of Elkin-1, so perhaps there was some developmental effects, and that's why they lost mechanosensitivity. To see if that is the case, what I did is I cultured the Elkin-1 knockout neurons, and then I put Elkin-1 back into these neurons using adeno-associated virus specific for sensory neurons called the PHPS vector. So, and then I performed poking with patch clamp. So first, I saw that indeed this viral mediated technique was efficient in transducing the sensory neurons. So this is a Elkin-1 antibody staining, showing that indeed Elkin-1 is now put back into this Elkin-1 knockout neurons. And then upon poking with patch clamp technique, I showed that indeed these large neurons now regain their mechanosensitivity. So this blue bar here is Elkin-1 knockout neurons with mock viral transduction, where you see low amounts of mechanosensitivity or mechanically active large neurons. And then when Elkin-1 was put back in, the percent of large neurons responding to mechanical forces has increased. This was very cool. So hopefully, so far I have shown you that Elkin-1 is required for normal mechanotransduction underlying touch in mice. This is basically the a story that I was presenting at the Keystone conference about 2 years ago now, where I met Amy, who was a doctoral student at that time in the lab of Mirella Dottori at the University of Wollongong. And she was actually working on human, like derived human sensory neurons, and looking at their mechanical properties, so looking at if they can produce indentation-mediated currents. So then we collaborated with their lab. So what they did is they induced human sensory neurons, and they knocked down Elkin-1 in these human sensory neurons, and then again performed poking with patch. They showed that after Elkin-1 was knocked down from these human sensory neurons, the amplitude of mechanically active current was vastly reduced in these neurons. So taken together, we can say that Elkin-1 is required for normal mechanotransduction underlying touch in mice and humans. So this was very exciting, but of course we wanted to go a bit further to say that does its function depend on other mechanosensitive protein? And the first thing we wanted to look at is piezo2. I won't go into too much details about this, but what I did was that I knocked down piezo2 in a Elkin1 knockout background neuron, and to show that piezo2 and Elkin1 share a role in touch transduction. So maybe I can go through this graph a little bit. So it might be slightly confusing. So the first two, black and gray, are control DRG neurons. So when I transfected the control DRG neurons with control siRNA, I saw that there was a large percent of mechanosensitivity in these large neurons, which is what we expect. Then when I transfected these neurons with piezo2 siRNA, I saw a massive reduction of mechanosensitivity, which is again what we expect, because we know piezo2 is a major touch transducer in sensory neurons. Then these two blue bars are for Elkin1 knockout neurons. So here, when I transfected the Elkin1 knockout neurons with a control siRNA, again, I saw less mechanosensitivity, which fits well with what I have shown you so far, that Elkin1 is an important touch transducer in sensory neurons. And then very interestingly, when I knocked down piezo2 on top of an Elkin1 knockout neurons, I saw that the mechanosensitivity was even less when compared to either piezo2 knockdown or Elkin1 knockout. So this suggests to us that piezo2 and Elkin1 plays a shared role in touch transduction, and like we can talk about this later on, but the idea is they're both expressed in the large neurons. So it's possible that some neurons are preferentially relying more on piezo2 or on Elkin1. Okay. So then, to summarize this part, Elkin1 is a novel mechanosensory ion channel necessary for normal touch sensation in mice and in derived human sensory neurons. So I actually have not shown you too much data on the ion channel stuff. So maybe I will take one slide detour on that. So, and I didn't go into too much details because the jury is still out on this, and this is because in 2022 there was a paper that said that Elkin1 is not an ion channel. It's Hoyle et al., and this is because they couldn't see a clear ion conducting pathway. Then in 2023, though, another paper was able to show a clear ion conducting pathway, and it's possible that this paper was not able to see it because this pore might have been blocked by lipid. So another very interesting thing that Han et al. Showed was that the Elkin1 ion channel has quite a bit of leak current, and this they found at minus 150 millivolt. And then upon mutation of some of the residues, they were able to abolish this current. So I actually was able to replicate this finding. So what you see here is at minus 150 millivolt, I found a bigger leak in HEC cells when compared to mock transfected ones. Furthermore, the same group did a more involved classification where in a proteoliposome they put Elkin1 ion channel, and they were able to show that at minus 150 or plus 90, so either very positive or very negative potential, this ion channel has very clear open and closed states. So this all gives us a very decent evidence that Elkin1 is an ion channel, and we have shown that Elkin1 is necessary for touch transduction. Okay, so then the second point I showed you was that Elkin1 mice show reduced paw withdrawal to cotton swab and other physical forces. Mechanoreceptors from Elkin1 mice show loss of mechanosensitive currents, and this is the neuronal basis of their lack of physical force sensation behavior. And then Elkin-1 function is independent of piezo2, but we think they share roles to regulate touch transduction. What I have not shown you due to time is that what's the role of Elkin-1 in pain neurons. And I think we are actively working on this, so we will come up with many other answers, I'm sure, and we will look into it in much more details. But just to give a very small summary, I found that Elkin-1 knockout does influence, it also reduces mechanosensation in nociceptors or pain-sensing neurons, but not to the large extent that it does in large neurons. But in general, pain is quite enigmatic, and we need to study that in much more details. And that brings me to the second part of my talk, where I will be using neuroproteomics to identify new proteins and ion channel which are clinically relevant pain targets. And this is very important because chronic pain affects millions of people worldwide, but it's very poorly treated. The three major drug categories are like anticonvulsant, antidepressants, and analgesics. Anticonvulsants like gabapentin, pregabalin, and antidepressants like duloxetine have very well-known central nervous system side effects. And opioids, as we all know, are highly addictive, and it's killing a lot of people worldwide, especially in the United States, due to opioid overdose. To top it off, actually, they do not produce adequate pain relief. So we need to do better at this front. And one of the attractive targets for us is the peripheral nervous system, because compared to the central nervous system, it's slightly easier to handle and manipulate. So for the rest of the talk, I will be concentrating on the nociceptor pathway, so the small and medium DRG neurons. The question, though, remains the same. What are the proteins that are involved in pain sensation in the DRG neurons? So as I was saying, that the DRG neurons are sort of an attractive target because they might be slightly simpler than the central nervous system, but of course it's not that simple. Using single cell transcriptomics, people have shown that there are over 10 different subsets of sensor neurons in the DRGs. And amongst this, but the good news is, amongst this, we sort of know which two are the pain clusters. So for the rest of the talk, you just need to remember these two cluster names: PEP, or the peptidergic cluster, NP, the non-peptidergic cluster. So these two are the pain clusters, classically speaking. So one can imagine that, well, if we know this, now we can induce pain in mice and compare these two clusters in transcriptomically between control and pain mice, and then we will come up with some good pain targets, right? And this is what people have been trying to do for over 10 years now. But unfortunately, we have not come up with a very clear candidate. And I think the reason for that is the proteomics gap. And what does it mean? It's the idea that the mRNA level and the protein level do not go in a 1-to-1 ratio. And there are many different reasons for it. The major ones are temporal disconnect, so they do not peak at the same time. There may be spatial disconnect, especially for such a long neuron like the DRGs, which innervate the peripheral periphery of our body. There can be the silent nucleus problem or the proliferation status problem. So the basic take-home message is proteomics gives you the current cell state when you're doing the proteomics, whereas mRNA probably gives you more of a steady state level of these cells. So this is very clear that we need to identify subset-specific proteome of sensory neurons if we want to find a new drug candidates. Unfortunately, this is now possible thanks to the ultra-low input proteomics method, and I have been collaborating with Fabio and Kasia at MDC on this. So this is possible mainly due to technological advancements. So we are using robotic delivery system to transfer samples. With this, we can manipulate about 300 picoliter of material, and also there is a technological development in mass specs in general. Now we have the ability to do single cell level proteomics. So with this technique, I wanted to ask a very simple question as a proof of concept: is can we quantify subset specific proteome in the sensory neurons? So this is how the workflow looks like. So I cultured the DRG neurons, and then I stained them with IB4 or TrkA. Why these two? It's because, remember, the two pain clusters. So IB4 corresponds to the non-peptidergic cluster, whereas the TrkA corresponds to the peptidergic cluster. And we also have the large neurons as a cluster because we are interested in mechanosensors in general. Okay, so after that, I, with laser microdissection setup, I cut out 50 of these in each category, much like Luke Skywalker, as you can see here. So, you can call it the lightsaber neuroproteomics. So there we go, the manual cutting. And then if it doesn't fall, you come in with the laser again and push it in. So once we collect these in these plates, we then process it using the mass spec. So then what's the result? What I found was that using these 50 neurons that I collected in each of these, Fabian's lab was able to quantify over 5,000 proteins in each of these categories. And so just to give you, give you a background on how it compares with the state of the art, this is the state of the art, is that from 12 whole DRGs, so this is DRGs taken out from mice with the muscle part and all other non-neuronal cells. So with 12 whole DRGs smashed together, where you lose a lot of the specificity, they were able to quantify about 4,000 proteins. So this kind of shows our technique is very, very efficient from the current state of the art. And we also wanted to compare how many of these sort of overlap, how many of these proteins overlap with the state of the art. And we find that about 3,500 proteins are similar, like same between our studies, which is good. We don't want sensory neurons to be vastly different from one lab to another. But in addition, we were able to quantify 2,500 extra proteins, and amongst these were hundreds of transmembrane protein, which we know are notoriously hard to quantify. So this sort of tells us that we have a technology where now we can find subset-specific proteome. And to look into it a bit more details, we did a PCA plot to show that indeed we can see the different clusters manifested here. So you can see the peptidergic and the non-peptidergic population quite separate from one another. This is now a volcano plot looking at the difference between IB4 negative and IB4 positive populations. If you're unfamiliar with volcano plot, what this means is these proteins here are upregulated in the peptidergic population, so TrkA positive and IB4 negative population. And if you are into peptidergic neurons, you can see many familiar faces here. For example, TRPV1, which is very well known, the capsaicin receptor, hot chili pepper receptor. Calca here is another name for CGRP, and anti-CGRP is an important drug for migraine, which is currently in use. So what we see is indeed all the peptidergic proteins that are well known are located in our peptidergic side of this volcano plot. So this gives us the proof of concept that we can define subset-specific proteome in sensory neurons. So now that we have this ability, I think we can now do a lot more with it. For example, we can now try and integrate multimodal information from sensory neurons. So we can do electrophysiology and then find the proteome of these neurons. And this is where I'm moving towards. So this, I will give you a very short kind of a taste of what we want to do with it. So the first thing is to find a novel pain targets. So for that, what I did is I generated this in vitro inflammatory model, and this was shown before that it works in rats, but this was the first time that I showed that it also works in mice. So when neurons are incubated overnight with NGF and PMA, what you see is the neurons become hypersensitized. So what it means is when I perform poking with patch clamp, I see larger mechanosensitive currents in these inflamed neurons, and also the amount of residual currents are increased. So both of these are indications of increased mechanosensitivity. And this is what's quantified here. So number of neurons with residual currents are increased, and the residual current amplitude themselves are increased. So once I phenotype this neuron, this is phenotype-driven proteomics now, I can take these neurons and then perform proteomics on them. And here I zoomed in only in the peptidergic subgroup because these are the ones vulnerable to this inflammatory insult. And with that, we are already, from this very small proof of concept study, we were already able to identify quite a few upregulated proteins in the inflamed side of this volcano plot. And remember, we are just keeping these neurons just overnight for like 8-9 hours. And even within this time, we were able to identify important protein targets. And now we're in the process of characterizing these to see if we can reverse the hypersensitivity induced by inflammation. Right, so all of this so far was very clinically driven. I sort of honed in this idea that this workflow combining electrophysiology and proteomics will give us an idea into how we can identify new clinically relevant proteins and ion channels in pain. But I just want to end with something slightly different to show that I think this technology has even further opportunities to give us insight into basic pain biology, and with the limited amount of tissue available. For example, from the sharks. Here is the new animal that Yelena talked about a little. So this started off as a very coincidental collaboration with a lab in Bonn. The sharks were actually growing too big, so they had to get rid of them. And they said if we were interested, and we said of course. And we sort of went down there to collect some sensory neurons from these sharks. And the question was very simple: do sharks feel pain? And why is this even a question? Is because there has been quite a few reports, and this has been going on since the 1950s actually, where people say that sharks, even when they're split into or disemboweled by other sharks continuing to feed, apparently they ignored their wounds. And so this kind of gave rise to this idea that sharks do not feel pain, and people then sort of take their fins off and throw them into the water, and all of these quite inhumane things continue. And just as a reminder, this kind of debate also happened in the fish field. But now we know a lot more, and we know that fishes do feel pain. But this has not been studied at all in sharks. So the first thing we did is we collected some nerves from the tail, from the fins, and we did electron microscopy. So before I show you the result, I want to say how it looks in a normal mammalian system where we know they feel pain, right? This is a cross-section of mouse nerve. What you see is there are some big nerves which are myelinated, so covered in this black substance. And in this SEM, although it's not super clear, you can see in between these, there are some other structures. So now if we look in bit zoomed in manner with TEM, what we see is these other structures are the unmyelinated fibers. So these are the classical C fibers or the nociceptors that transduce pain in mammals. So again, the big fibers which are surrounded by myelin are the touch sensors, and then the unmyelinated fibers, the pain sensors. So what happens then in sharks? In sharks, what we see is, again, the myelinated fibers are present. There are lots of nerves with myelin, but when you look at the structures in between, there is nothing. And then you can look closer with TEM. You see again that there are myelinated fibers of a variety of diameters, actually, and this is the interesting part maybe, but there are no unmyelinated fibers. So then I think the question still stands, do sharks feel pain? And just to kind of give you more insight, in 1993, Peter Snow showed that at the DRG level, he did not see a lot of variability in diameter, because remember I was saying so far, the large diameter neurons are touch sensors, the small diameter neurons are pain sensors, but what he showed here is that most of them look large or maybe small in from a relative point of view, but they are basically a unimodal distribution. But of course, we also collected DRG neurons, and I stained these DRG neurons now with CGRP. So this is one of the, this is what I was saying was one of the drugs against migraine. So it's the pain marker, if you will. And I see that there are positive CGRP positive neurons in these sharks. But of course, it's super hard to validate anything in sharks, right? And we don't have a lot of tissues. So I think this is where the power of ultra low input proteomics can really shine. So now we have these neurons, we can collect these neurons and do this ultra low input proteomics to show whether there are pain markers in these neurons, because it's quite plausible that maybe these sharks do not have distinct systems for pain and touch, but they are able to sense pain, and maybe they do not need two completely different systems. So these are all questions that are remaining. And I think if we come in with this workflow where we can do multimodal analysis of behavior, then electrophysiology, and then do proteomics, we are able to answer this very fascinating question of these very ancient animals about, do they feel pain? And if they do, what's the mechanism and how does it differ from our very classical idea of pain? And actually, during the Society for Neuroscience, Society for General Physiologist meeting last year in Woods Hole, I met Andrew, and he had some sharks in his lab, and we are actually collaborating on this because unfortunately the lab in Bonn closed. So hopefully we will continue this, and we will know more about sharks in the future. So I just want to finish with this idea that I think the pain physiology field will move more and more into this multimodality, where we can combine information from electrophysiology, behavior, and proteomics from same group of neurons, or maybe even a single neuron, and that will give us insight into the fundamental biology as well as give us clinical insights into pain physiology. With that, I would like to thank all the members of the Lewin Lab and Kasia Lab and my collaborators at Australia, Bonn, the funding bodies, and thank you very much for listening, and I will take questions.
Thank you very much, Sam. That was so interesting. We already have 4 questions in the chat. I will read them out now. For those of you who are unsure, just write your questions in the chat. That's how we do most of the questions. I will read them out and Sam will answer one by one. You can also talk, so just raise your hand if you want to. And Greg will then upgrade you from attendee to panelist because otherwise you won't be able to talk. Okay, but there's loads of questions in the chat, so let's get going. So first is from Anna Boccaccio, and she's from Institute of Biophysics Genoa, and she's asking how reproducible is the response to indentation or poking stimuli in DRG neurons? Is a single neuron always responding with the same typology of response when stimulus is repeated?
Yes, so with poking, it's quite stable. So if a neuron responds with a rapidly adapting biophysical property, at first, it keeps doing that for the entirety of the stimulus response curve.
Okay, thank you. So the next question is from Alison Obergrusberger. Very nice talk. Thanks, Sam. I was wondering if you know if Elkin-1 is activated by shear stress, i.e. fast fluid flow, as well as poking. Also, do you know if Elkin-1 is sensitive to temperature? Thanks.
So no, I don't know. Actually, it's possible. It's not super pressure sensitive. So we did some outside out patches, and in those the cells were not active. I did not see any currents, but we haven't tested shear stress, and I don't know at all about temperature. But we have some data at like in vivo level where we see no change in Hargreaves. So the mice do not, the Elkin-1 knockout mice do not have different behavior in hot plate, for example.
Okay. So the next question is from Kim Lacan. Thanks for the nice talk. Concerning the 4,103 proteins quantified in 12 whole DRGs, was this performed in human DRGs? If yes, what tissues are these neurons from?
No, those were from mouse DRGs. Actually, we are working with human DRGs, but no, that paper that I showed was from mouse, which that's why we compared it with our mouse dataset.
Okay. So the next question is from Damien Bell. Lovely talk, Dr. Chakrabarti. Combining the 2 halves of your work, have you combined single-cell proteomics with Elkin-1 expression and or current recordings to find putative Elkin-1 accessory or functionally coupled protein partners?
I think that's a great idea. No, I haven't done it, but we have the data from the large neurons and we see robust Elkin-1 expression. But I guess to find like functional and structure coupling, we have to do a bit more. We have to do these TMT labeling or something like this so that we can pull down Elkin-1. I guess that's where we have to do in the at a later stage. But no, we haven't done that.
So the next question is from Mari Cronin. Thanks, Sampurna. If in your Elkin-1 knock-ins EpIC experiments, what is your operational definition for a mechanoresponder? Indentation depth, required current magnitude, or kinetics?
So I guess this question is about the human induced neuron experiment. And for this set of experiments, basically, if there was a reduction in mechanically activated currents, we defined it as a reduction in mechanosensation. But of course, one needs to do more and maybe even record from primary human sensor neurons with Elkin-1 knockdown to see what happens there. But this is an initial proof that even in human system, Elkin-1 knockdown reduces mechanosensitivity.
Okay, so we have another one from Kim Lacan. Concerning the shark nerve fiber composition, the A delta fibers slightly myelinated also transmit noxious stimuli. What do you think about those?
Yes, I think it's very likely that the A deltas are transducing noxious stimuli, but it's just super fascinating that there are no C fibers. But definitely the A deltas will play a role, and it is possible that the A deltas have expanded. Like all of them are now A delta or A beta. That's very, very possible. We are actually looking more into this myelination idea, and we see quite interesting things like it's maybe that the A betas or the big neurons have more myelin instead of like the smaller neurons having less. So yeah, it's possible.
Okay, the next question is from an anonymous attendee. Thanks for sharing your work. Did you also try AAV PHP as the TD tomato empty cassette for your poke and patch experiments? And the next one, when you say that transcriptomics gives us a steady state level, are you suggesting that there is no effect of external stimuli?
So for the first question, yes, we did actually. Let me go back. So the mock was Elkin-1. The mock part was basically just the AVPHP with no Elkin-1, but with TD tomato. This is the second. So this one here, when I show this mock here, this is essentially AV PHPS with TD tomato, so no Elkin-1. And then the second question was, no, of course, there is external stimuli that affects the steady state, and we can also see it at a transcriptomics level, but I think the time course is very different. So to see the changes at a transcriptomics level, one probably has to wait much longer. The fast changes is something that you can't see in transcriptomics, and this is where the strength of proteomics comes in.
So the next question is from Mark Rogers. You used Usokin's transcriptome data, but this is from rodents. Have you looked at more recent human DRG and IPCS sensory neuron transcriptomes to see which types of human sensory neurons express Elkin-1?
Yes, we have looked into those data sets as well. And again, from a transcriptomics data set, it seems like Elkin-1 is expressed in most of the clusters, actually all. But I think in protein levels, it might, in the proteomics level, it might be slightly different. There might be more or less in specific subgroups, which I think one needs to do.
Okay, so the next one is from Artem Kondratsky. Thank you for this insightful talk. The shark part is very interesting. So we have a bunch of different mechanosensitive ion channels, Elkin-1, piezostrax, OSCA, maybe even TCAN, well, some TRIPS, A6. Why do we need so many different mechanosensitive ion channels? Do they detect different levels of touch like TRIPS do for temperature?
Yeah, I think this is a question that's super interesting, but it's not surprising, I think, to me, because if you look at the other sensor system or biology in general, there is a host of redundancy, and I think we need the redundancy, because if something goes wrong in one, there needs to be someone else who takes up the job. So I think it's very necessary, and of course they might also sense difference in levels, because as we know, pain and touch is also a difference in level. So the same stimuli, if you give it at a higher level, it becomes painful. So yeah, but I think the redundancy idea I like a lot, because it's present everywhere in biology.
I just have, that will be the last question in the chat for now. There's one technical question, maybe Greg can address that. But yeah, I think attendees can only type questions in Q&A speech bubble rather than the chat, from what I'm aware of. But I was wondering if I can just squeeze in one quick question. So
Yeah.
you said that Elkin-1 transfected cells have a high leak current.
Yeah.
So is that, do you see that in neurons as well, or is that only when you transfect HEC cells?
So this I haven't done in neurons, right? But this would be a good idea to do between Elkin-1 knockout versus like normal wild type mice, right? This is what would be an idea of doing. But with neurons, leak current quantification gets slightly complicated because of a lot of voltage gated ion channels. Yeah.
Right. Yeah. It's a bit trickier. And maybe just one quick one. So
Yes.
for the when you were working with mice and Elkin-1 minus mice, you could see these rapidly adapting, intermediate adapting, slow adapting currents. And in your experiments with human iPSCs, could you? I know you didn't go into that sort of analysis, but could you distinguish something similar or not?
No, actually, these neurons that Amy and Mirella's lab differentiate, they're all rapidly adapting. These are made in this way that they produce mechanoreceptor-like, they're mechanoreceptor-like neurons. They're all rapidly adapting.